not at all. We have already covered many chapters of the class 12. We have covered reproduction in human being. Then we have covered the reproduction and sexual reproduction in flowering plant. We have covered reproduction in reproductive health. We have covered the principle of inheritance and variation. So now today we are standing at the sixth chapter, that is the molecular basis of inheritance. So go through with the molecular basis of inheritance in case if you have any problem in any of the heading or any concept. Because you people are already going through the 12th syllabus, so I am not going through with the syllabus. I'm just dealing with the previous year question paper and questions.
Sir, can you show the questions of this only? I'll take a screenshot. I was not present, sir.
Question of no, we are going to start this chapter today. Molecular basis of inheritance. So what I said.
Okay, sir. What did we do in last two classes, sir?
Last two classes we did like we have done in last classes inheritance of variation and that part that human health part and that last to last part human reproduction part and plant reproduction.
Okay, sir.
So today go through with the molecular genetics part. Take your NCRT, go through with the molecular genetics part. In case if you have any doubt in any of the point, then we can discuss that, right? Otherwise, the conceptual part is taken care of. Yeah, hello, Rabia. Rabia, I think you was there in last class.
Hello, sir. Yes,
Okay.
I was there in the last class.
Yeah. So we have in last class we had completed till the first chapter of genetics. That's the inheritance and variation part, right? And today we were supposed to start with the molecular genetics part, right?
Yes, sir.
Okay, fine. So go through quickly, go through with your molecular genetics part, right? Go through with this chapter. In case if you have any doubt in any of the concept, then I'll discuss with that. Then we will start with the previous year question paper and then rest of the question. Let me know when you complete it, right? Hello, Aisha. Aisha, do you have any doubt in this chapter, molecular basis of inheritance?
No.
Fine. So, did you go on through with it, Rabia and Afia, or are you still going through with it?
Sir, we'll start, sir. If any doubt I have in between, I'll ask, sir.
Fine.
Because it's so much is there, right?
So much is there. You people have already gone through with that in detail,
Yes,
right?
sir. I
So
know
you
it.
just need to
So
go through, just had a glance with that. Rabia, what's about you? Hello, Rabia, am I audible to you?
Yes, sir.
Yeah, what's about you? Did you go through with that?
Yes, sir.
Fine. So have any doubt?
No, sir.
Fine. So let's start with the previous year question. Look, yeah, Rabia was there. So I told even Aisa joined many classes. So as I told you people that genetics is the core of 12th class syllabus for NEET examination. And average you will get 18 question out of it, 17, 18 question out of the whole syllabus. So first question is DNA fragment R. So I have already that circled. So how they get the negative charge? These are all previous year questions, like they are asked questions. So how do they get negative charge? A very simple question. This is in their your NCRT. No idea? Look. DNA, what is the composition of DNA? So when you say the composition of DNA, so DNA is made up of nucleotide pairs. What is the composition of nucleotide?
Sir, it's because of deoxyribonucleic acid, sir. So because of the acid, it is negatively charged.
No, no, no. That is not a logic behind. Right? Why this is acidic? That's why it is negatively charged. Look, nucleic acid. So nucleic acids are made up of nucleotide, right? So they are
Yes,
made
sir.
up of nucleotides. So what is the composition of nucleotide? Uh, okay, fine, Aisa. Pedigree. Was you there in last class? I discussed pedigree in detail with Rabia. Was you there in Aisa last class? I think you was not there.
Sir, pedigree, can you discuss again, sir?
Just a minute, just a minute. Aisha, was you there in last class? Pedigree, I
Was
discussed.
there on the Tuesday class, but then we didn't discuss.
You was there in last class.
Yes, sir, but I did
No, I
not.
discussed. Rabia, do you remember? I discussed in detail. Even I
Yes,
discussed the
sir.
question. Yeah, I discussed
You did
question.
pedigree.
Yeah, pedigree. I discussed in detail. Like I discussed color blindness. I discussed this one, hemophilia. Along with the, in fact, I discussed with a question. I discussed a question with Rabia in detail. If you'll go through the video. Anyway, at the last of this class, I'll tell you again. I'll revise you. I'll give you a recap. Right? No issue. So first, complete this chapter as we have started. So nucleotides are made up of three components. What are those components? So major component is sugar part, right?
Phosphate.
And then nitrogenous bases, right? Phosphate part, right? So phosphate, this phosphate part, this is negatively charged, right? And this is basically phosphoric acid, H3PO4. H3PO4, I have shown over here, it is negatively charged. So outer side of the backbone, the DNA is a coiled particle. Outer side of the backbone is made up of phosphate part or phosphoric acid, right? And phosphoric acid is negatively charged. So because of this phosphoric acid, because the repetition of question is not possible, right? Normally questions are not being repeated, but you get the questions from these questions, right? You may get because of DNA is negatively charged because of sugar part, nitrogenous bases part, phosphoric acid, or like this way, right? So because of this phosphoric acid part, DNA is negatively charged. Got it?
Yes, sir.
Second question is very beautiful question, and this kind of the questions are frequently being asked. The question is, if there are 999 bases in RNA that code for a protein with 33 amino acids, and the base at position 901 is deleted, such that the length of RNA become 998. How many codons will be altered? Can you tell me? Very simple question, but logical one. So what
Sir,
kind of
61 codon is triplet, so 61 codons will be there for sure. And plus 3 will be there for which does not code for any amino acid. So total of 64 codons will be there, sir. So when we minus it, it will be
no.
no, no,
No,
sir.
no. This is a very basic concept. You are not getting the question. In fact, read question again. Read it carefully again.
If there are 999 bases in an RNA that codes for a protein with 33 amino acids, and the base at position 901 is deleted, such that the length of the RNA becomes 998 bases, how many codons will be altered? So one set of codons will be altered, 3.
No. Anyone else? Rabia? Any idea? Aisha? What about you? Look, It's very simple, very simple. Try to understand. First, can you tell me what kind of this mutation is, or what kind of this abbreviation is? Any one of you, come on, tell me. This is a mutation. What kind of mutation is?
Point mutation, sir, because only one is deleted.
This is not point mutation. Point mutation cannot be recognized. Point mutation is that one, suppose that if get altered, not deleted or added, that will be point mutation. And point mutation can change only just one codon or one amino acid. That's it. And normally point mutation are very frequent kind of the mutation, and they cannot be seen. They cannot be seen in our body or in any animal or any plant. Look, try to understand what I am saying. There are 999 bases. Fine. So there are 999 bases. Let me make you clear. So what question is saying? Total there are 999 bases. Out of this, and this is a RNA, leave it. 33 amino acid, it have nothing to do with it. Base at the position 901. Suppose that this is 901, right? And this is 999. If one will be deleted, obviously it will become 998. After this, if one is deleted, if one nitrogen base is deleted, right? What is that? At the position 901 is deleted, right? The base. So one base is deleted. Suppose a thymine is removed from here, or sorry, uracil is removed from there. So what will happen? Now the frame will shift. The complete codons will get altered after 901, right? So how many codon will be altered after 901 till the 999? All will be altered. So here 901 upon 998, total 98 base pairs will be altered, right? So 98 bases there are between 901 and 999. There are 98 bases. 98 bases will get altered, and 3 base make one codon. So 98 upon 3. So what is the answer? 33. Total 33 codon will get altered. That's why their answer will be D. This is frame shift mutation. Did you got this point or not? Tell me.
Yes, sir.
Rabia and Aisha. Tell me frankly, if you got this concept, it's okay. If not, I'll tell you. Do you people clear with what is the frame shift mutation?
Yes, sir.
Excellent. Right. So this is frame shift, and you got the answer of the you got the answer. How I
Yes, sir.
calculated? Very good. Excellent. You will get this kind of the question, right? One more thing. Suppose that if question says not deletion, if question says one nitrogen base is replaced. Now, what will be the answer?
Same answer. Nothing will be changed. It's just replaced, right?
Yeah, no. There will be something will change over there. How many codon will be altered? At least
3.
one will be altered. One will be altered. The answer will be 1. You have replaced. Suppose that here was a codon AUG. You have replaced U with C, right? Now, what will be the codon? Codon will be ACG. So obviously, one codon will be changed, right? So you will get these kind of the question. So these are the sample of the question. You may get same kind of the question, and this kind of the question have been frequently seen, right? So practice like this kind of the question. Which of the following RNA act as structural RNA as well as a ribosome in bacteria? This is a factual question. 23 sRNA. Did you got it or not?
Yes, sir. It's written in the book, sir. It was written.
Very good. The equivalent of structural gene is, so tell me what is muton, what is operon, and what is cistron? Do you have
Sir,
clarity with
lac
all this?
operon is there, sir.
Okay, so what is operon? Well, lac operon is lactose operon. That is a model, right? So what is operon
Yes, sir.
basically?
Operon is like it is like many cistrons together. It will be it will be a it will be operon, sir, in a arrangement.
Rabia, Aisha, what is the operon?
Sir, it has promoter and regulatory genes.
Right. Now you got the point. Operon is made up of, look, a regulator gene, operator gene, and a structural gene, right? This part is called structural gene. This part is called cistron, right? So operon is made up of regulatory gene, operator, promoter, operator, and structural gene, right? Again, come to this. So operon is made up of these components. Muton, what is muton? The gene where mutation take place is called muton, right? This was muton. The gene where mutation takes place is called muton. Cistron, this is equivalent to structural gene. So structural genes are known as the cistron, right? So this is also a factual question. This was not a logical question. Twyler conducted an experiment to prove semi-conservative model of the chromosome replication on E. coli, right? So everywhere we have done the experiment with the E. coli. Which of the following RNA is the most abundant in animal? Ribosomal RNA, right? rRNA is the most abundant. Which is the least abundant RNA?
tRNA.
tRNA, right? tRNA is the least one. Now, the association of H1 with nucleosome indicate DNA is condensed into chromatin. So, what is the role of H1? Who will tell me? What is the role of H1 protein? What is the role of H1 protein? Quickly, come on. You have already packing of DNA part. You must have gone through with that. You must have studied packing of DNA.
Sir,
And how many type
there is
of
no there is no description about H1, right? Only histone.
how many type of histones are there? How many type of histones are there? Rabia, Aisha,
Sir, there are 8.
come on, not 8 type. There are octamer. How many type? Only 5 type. H1, H2A, H2B, H3, and H4. Look, what is the role of H1? H1 hold the DNA on octamer. It do not allow them to slip down. Let me recall you. Packing of DNA. What is the packing of DNA? Look, this is octamer. If you want, you can take this screenshot because these are the frequently asked questions. Look, the core of octamer is made up of 8. That's why we call them octamer. The core is made up of 8 histone molecule, and every histone molecule have 2 copy, right? So H2A, H2B, H3, and H4. Look, H2A here, then this is the copy of H2A. H2B here, downside there will be another H2. H4 here and H3 here. So core is made up of 4 kind of protein. H2A, H2B, H3, and H4, right? So total octamer 8, right? This H1, then what happened? Then this DNA wrap around this octamer. We call it nucleosome. Now, this DNA can slip down from octamer. So it do not get slip from octamer. It is sealed and packed with the help of H1 protein. So how many type histone are there? Are 5 type histone: H2A, H2B, H3, H4, and H1. Right. Between two nucleosome, there is a DNA which we call linker DNA. Linker DNA is a DNA that connect. Linker DNA is a DNA that connect one nucleosome from another nucleosome. Right. Now, what is the length of a nucleosome and linker? Right. So length of a nucleosome and a linker DNA. This is made up of two-handed nucleotide. If you want, you can take the screenshot. This is very important. Did you got it? What is the role of H1?
Yes, sir.
Okay, take the screenshot quickly. One more question. Why without histone? Why without H1? Without H1, DNA wrap around the histone protein. Why and how? Which force help DNA? Let me make it easier. Which force make DNA to wrap around histone protein? All of you, quickly. Very simple question. This is a basic question. Very simple.
Sir,
Why?
it's actually it's positive and negative, so they attract each other and
Very good.
they
Very
bound
good.
it, right?
Very good. Excellent. Right. Histone protein is a positively charged protein, and DNA is negatively charged. We discussed, and because of this opposite charge, due to electrostatic force, they wrap around it. Right. But this is not the question for NEET examination. It's a simple concept. That's why I said it's basic concept. Which component give positive charge? I told you which component gave negative charge to DNA. Now the question, this has already been asked in AIMS examination. So old question, right? But it can be asked again to you in NEET examination, right? Which component give positive charge to the histone protein? Like phosphoric acid give or phosphate give negative charge to the DNA. So which component give
Sir,
positive charge?
it is lysine, arginine. They
Okay.
are all they are present in also here H2B and H4. They give positive charges, sir.
What are they? Lysine. What are the lysine and arginine?
They are a type of histones, sir, present in type of histones, so they
No.
give
The histone
positive
is a protein,
charges.
and protein is made up of amino acid. Right. Very good. Lysine and arginine amino acids. Please note down lysine and arginine amino acid. They give positive charge to histone protein. They are found in all. Right. They are found in all H2A, H2B, H3, H4. Right. Lysine and arginine are rich in histone, and they give positive charge to histone protein. Right. Great. Come to the next question. The final proof for DNA as a genetic material comes through the experiment of MacLeod, McCarthy, and Ovary experiment that given that just a minute. Final proof for DNA is a genetic material. Right. This is the Avery, MacLeod, and McCarthy. Fine. Now come to the next question. Spliceosomes are not found in the cells of. Okay. What are the spliceosomes? Spliceosome.
They are used for splicing, sir.
Right. Splicing of. They are used for splicing of.
Splicing of mRNA.
Right, very good. mRNA. Look, transcription. Maturation list. Look, maturation of mRNA. So this is a mRNA. Every mRNA have exon and introns. Introns are useless or non-coding part, right? Exons are coding part that code for protein. Now what happened in an intron region, this splicing protein comes, right? This splicing protein, they bind like hairpin loop, and these structures are spliceosomes, right? Spliceosome, splice of intron means non-coding part, right? And then after that, the coding part unite with each other, and they are this is called maturity, right? So spliceosomes are, they are made up of what? The splicing protein and plus non-coding part or introns. Got it? And this structure is called hairpin loop of introns. Okay. Great. Now one more question. The commonly used vector for human genome sequencing is, this is given in Human Genome Project, BAC or YAC. Is that clear? That we have done. Uh, The answers are given over there. You can go through with it after the class. And before that, you were asking about the pedigree analysis. Let me recap you again. Pedigree analysis. Uh, Pedigree. Look, pedigree. We did the questions. This question is there. Yes, that is already open. So I think you people must be clear with the color blindness and hemophilia. Yeah, and Aisha. Yes, Aisha.
Yes.
Okay, fine. So you both are aware with the hemophilia and color blindness. So let me first tell you what is the pedigree analysis. So pedigree analysis is just a type of use of the symbol. We can tell after what generation how the genes travel from one generation to another generation, right? So look, in pedigree analysis, these are some symbols. So this is square is meant for male, that is unaffected, means do not have disease. This solid block is male affected with this disease. Female unaffected, male female affected, female unaffected, right? Now here the unknown sex or sex unspecified. What does it mean? Suppose that I said that there is a color blind female and a normal male, and they have got three child, right? Or they have got three offspring, right? Now when I say there are three offspring or three child, you sex is unspecified, means all three can be boys, all three can be daughters, all two can be daughter, one son, there can be two daughter, uh, one son, and two son, one daughter. So any kind of the combination is possible. That's why we will say, so we will make this symbol that is unknown sex or unspecified, right? This is the carrier female. They carry the genes, but they are not affected by disease. So normally, not normally, always females are carrier and males are affected. Males cannot be carrier, right? Then they are unaffected individual affected. This line shows, this horizontal line shows mating or marriage, right? Double line, they show mating between relatives or consensuous mating, right? Means selfing, fine. This slash means death of the individual. This is male and female, they have got a male child. This is affected. Male, female, normal male and female, they have got a female child with affected with disease. Male, female, one male child, one female child, and both are unaffected. Again, the same, right? So these were the symbol. And let's talk about the pedigree. how the pedigree questions are being asked. So, the question. Right. So here there is a question. A normal vision man whose father was colorblind. So a person have normal vision means what should be the genotype? Means he do not have any disease. So I will make this box, right? And X and Y, right? Males are not carrier. He's a not colorblind. So a normal vision man whose father was colorblind. It have nothing to do with his father. If he have normal vision, it means he do not have what? Color blindness. Fine. So a normal vision man. So this is a person who have normal vision, right? X and Y. Marries with a woman whose father was also a colorblind, right? So if father was colorblind, what will happen? If father was colorblind, what will happen? What kind of female it
Ma'am,
will be?
the father is colorblind. It does not matter. He will not be a carrier.
No, 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 no. If a father is colorblind, she will be carrier. Look, X, C, and Y, right? His father is colorblind, right? Mother, it is not there, so we can take normal mother. We will take normal mother, XX. So look, XC, X. She is a carrier. Come to another daughter. Another daughter, X, C, X. Carrier. What about the sons? Sons, both of the son will be normal, right? XY. Another son will be normal, XY, right? So when a question says that marries with a woman whose father was also colorblind, if his father was colorblind, if her father was colorblind, it means it means daughter will be colorblind, right? So what can be the combination? The combination will be like this way: XC and X. Now they have got their first child as a daughter. What is the chances of child would also be colorblind? Their first child is daughter, right? So take make the chart, right? What can be the combination? So
So,
let's take
so the daughter will be only carrier.
exactly
Carrier. It
XC.
won't be affected.
50% daughter will be carrier, right? XC, then X. So 50% daughter of the carrier. What chances that child would also be colorblind, right? The first child is daughter, there is a 50% chance. Did you got this question or not?
Yes, sir.
Rabia and Aisha, what's about you?
Yes.
That's how we have to do the question, and that's how, look, this question belongs to 2012, and still they are asking that continuously. The questions are coming like this way only, right? So you will get the question from pedigree analysis, and pedigree is very important, right? Now again I would like to say, you go through with your this one, molecular basis of genetics, right? And ask me if you have any doubt, because this is a very important part. All of you, Rabia, Aisha, and Fia. DNA fingerprinting, yes, you will get the question. Human Genome Project, yes, you will get the question. What kind of the question? And DNA Human Genome Project, which is the largest, which is the chromosome? Tell me the chromosome which have the largest number of the gene. The chromosome have the smallest number of the genes. That kind of the question. In DNA fingerprinting, the dye have been asked twice. Which dye we use in DNA fingerprinting? And what color it gives? Both question have been asked. The dye which we use in DNA fingerprinting, what color it give? It is also mentioned in your uh, biotechnology part. Come on, ethidium bromide, it give orange color. DNA fingerprinting, lithium bromide, it gives orange color, right? Total number of gene nucleotide 3164.7 million, right? Average gene is made up of 3000 base pair. That kind. Longest gene is diatrophin. Yes, this question I've been asked. Longest gene is dystrophin. It have 2.4 million bases, right? Go through with this chapter and ask me if you have any doubt. And if you do not have any doubt, say it's clear to you, then I will move to the next chapter. Come on, quickly. Pia, Rabia, Aisha, are you going through with that?
Yes, sir.
Evolution, I'm not sure this year it will be in syllabus or not. But yes, there are some chapters which are important in evolution aspect only. So done? Go through with that?
No, sir, no.
Rabia, what's about you? And Pia, what's about you? In next class, we'll discuss two chapter. We'll try to discuss two chapter. First, evolution. It's a big question chapter, but there are few questions or few important part from where the questions are coming. Human health and disease, again the same thing. Uh, Not too much question, but yes, the question come from the disease, from the pathogen, from the vector. So the factual question you will get from here. And evolution, again, some example based question will be over there. And strategies for enhancement of food production, hardly there will be any question from this. Hardly. Microbes in human welfare, again the factual part, right? So these are the chapter from where the factual questions come. There is no conceptual part. And the all four chapter include a bit small relevance for NEET examination. But yes, sometimes they do ask question from. We cannot deny them, right? So we'll discuss in these four chapters. So what I would like to say you go through with the evolution, go through with the human health and disease, strategies for food enhancement, at least next three chapters, and we'll discuss the questions and concept of all these three chapters. Okay? And ask me if you have any doubt. Any query, any question, any doubt from any of the topic. You're most welcome. Rabia and Aisa, no doubt.
No.
So please go through with before joining the next class, right? Go through with. I hope that you will. If you will go through with it, right, and you will note down your doubt. This will be easier for you, right? Got it? Okay.
Yes.
So see you in next class, right?
Thank you.
Yeah. Bye bye. Good night.